Movie. This is November 19th. And folks, today is the last day to drop, okay? So if you're thinking maybe this just didn't turn out to be the right time, right semester, just remember that today's the last day to drop. And if any of you want to touch base or talk, please do. Also, folks, this Friday is an open lab. And I changed the time, folks, to earlier, 8 a.m. to 10 a.m. because something else came up that I have to go to on Friday. And then, folks, do remember that a week from today is our lab exam 2. And I will post on Canvas the start times. For most of the lab sections, we'll be starting a little bit later than normal. If you have accommodations, remember you're coming early. And also, folks, and we talked about this, we'll talk about it in lab, you're going to be turning in your lab exam 2 study guide packets, and we have a checklist in lab. I'm going to ask you to also turn in your lab observations, any lab results you made. You don't have to rewrite anything. You can just staple lab observations to your packet, right, so you get credit. And if in doubt, you can, if you're like, I'm not sure if this lab observation is important, the more you add, the better. Like if you add additional observations that aren't on the checklist, you might be able to get some bonus points, right? So if in doubt, give it to me, right? That's kind of the way to get the maximum number of points. But again, we have a checklist in lab, folks. So in the 1 p.m. lab, we'll be discussing that. Okay, so folks, last time we had finished up viruses, but I heard on the radio something that I thought was really interesting. They were talking about the polio vaccination, and the World Health Organization, folks, has made it the goal to try to eradicate polio from the wild, from nature, just as they did with smallpox, right? But it's been really tough. There's been some big issues. And they were sharing that of the 3 strains of polio, they've eradicated 2. So 2 have been eradicated from the wild. But the 3rd one is still circulating. And the heartbreak is the vaccine they're using. In many parts of the world, you guys are using the OPV. What does OPV stand for? The oral polio vaccine. Is that the killed or the attenuated live? It's the attenuated live. The OPV that they're using, you guys, it's in some cases reverting to virulence. What do we mean reverting to virulence? So you guys remember this OPV, it's the attenuated live. Attenuated meaning weakened, it shouldn't cause disease. So what do you think reversion to virulence mean? What's the nightmare with these live attenuated viral vaccines? What don't you want them to do? Is become virulent again, right? And you guys, what's really disturbing, they reported that the OPV they're using right now, when it reverts to virulence, it's hypervirulent. And you're like, what does that mean? It's far more aggressive in invading the nervous system. Isn't that just a heartbreak? Oh my God. So we could say that it's hypervirulent. So there's increase in nervous system infections. And that is such a bummer. So you guys, if you had unlimited money and unlimited people to carry out vaccination, what would be one solution? Do you have another polio vaccine that might be safer? Which polio virus do you have another polio virus vaccine that might be safer? IPV. What's IPV, you guys? Exactly, right? So if you had unlimited money, unlimited people, right? A solution might be to go back to the inactivated polio vaccine, like what we use here in the United States, right? You guys remember IPV stands for, what was that? The inactivated, the killed, right? So remember with the inactivated polio vaccine, you guys, is there any chance of it reverting to virulence? No, it's dead, right? Right? So it was kind of, you know, like as a teacher, I'm like, oh my gosh, this is perfect. You know, this reinforces what we were discussing last time, but also very heartbreaking that now with some of the OPV poliovirus reverting to even hypervirulence, now we're seeing even more cases of paralysis caused by the OPV. So you guys, who developed the IPV? What was the researcher's name? Salt, right? So sometimes this is referred to as a salt polio vaccine. Who developed the OPV? Sabin. Good job, you guys. All right. So I just thought it's like, wow, that was timely and such a bummer. Okay. All right, folks. Then we did leave viruses, and we had started looking at prions, right? And folks, I am so sorry. I forget where we left off in our prion presentation. Had we gotten to lack of an immune response? Had we gotten to that slide, you guys? That's really important. Shall we start there? And if we're past there, yell at me, okay? Because I know you don't want to hear the same thing twice. You guys, but this is so important. Our immune system doesn't recognize the misfolded prions as being foreign, right? So our immune system doesn't mount an immune response against the misfolded prions. And the reason this is bad, well, number one, there's no immune response that potentially you would hope could protect us. But secondly, you guys, this means that we don't make antibodies against the misfolded prions. And the reason antibodies are important, they're protective, and we'll talk about how they protect us in the immunology section. But secondly, you guys, antibodies are really important in diagnostic tests. Often the way we determine if a person has been infected with a pathogen is we look for antibodies against a pathogen, right? And it would be a perfect test if our bodies made antibodies against the prions. Like for me, they could take a blood sample and screen for antibodies against the prions and say, oh yeah, you're incubating it, right? But there's no such test because our bodies don't make antibodies. So this means, you guys, it's really hard developing a good diagnostic test for potentially infected hosts before they die, right? Usually, we might base a diagnosis on clinical signs and symptoms, but very often the ultimate is to look at the brain, look at the histopathology of the brain, and that's after the person or the animal has died. So the need for a good diagnostic test is really, really important. So folks, had we gone through, I think we'd hit scrapie, like just really briefly, scrapie, the first prion disease of sheep and goats, and then we talked about the discovery of Creutzfeldt-Jacobs disease. But did anybody know what was causing these diseases? Nope, nobody knew. These were prions. And then you guys, we mentioned Kuru and the Foray people of Papua New Guinea. Had we mentioned that? Okay, all right. And then had we gone through the Kuru, folks? How it initially evolved within the Foray people and how it got transmitted? Yeah. Okay, good. So we think it started with Creutzfeldt-Jacobs disease and then through mortuary cannibalism, right? It was spread from person to person. Good. And then when the mortuary cannibalism was halted, the number of Kuru cases went way down, way down. The only concern is, folks, is that it's believed that prion infections can have really long incubation periods, meaning we can be infected, we're not showing clinical signs or symptoms yet. And so this is always a concern with prion infections is like, is the person in that incubation period not showing clinical signs or symptoms? And again, folks, this was this outstanding Australian documentary on the history of Kuru. It's excellent. It's heartbreaking because you actually see the Kuru victims. Again, it's very sad. It's a really, really heartbreaking documentary. But if you have time, even after the course is over, this might be worth looking at because they draw many parallels to the mad cow disease, the variant Creutzfeldt-Jacobs disease that we still have worries about these prion diseases. Had we gone through the different types, you guys? Had we gotten down to the variant variant, Creutzfeldt-Jacobs disease, we haven't gotten there. Okay. So folks, just, and you wouldn't have to memorize these frequencies, but the prion diseases, Creutzfeldt-Jacobs disease specifically, there's about 1 case per million people. There's 4 types, the inherited or familial form, and this one's a tough one, you guys, it's autosomal dominant, so if you inherit the bad gene from one parent, you will eventually develop the prion disease, so about 10 to 15% of CJD cases. Iatrogenic is when you are infected through some kind of medical procedure. Iatrogenic literally means caused by the doctor. So these would be medical procedures where either contaminated medical instruments contaminated with the disease-causing prions are used on you and are introduced into your body, or you receive maybe tissue or pituitary extracts from a person that was incubating prion disease. So when you receive the tissues, the organ transplants, the growth hormone, you're actually receiving the misfolded prion, and then you go on to develop the prion disease. The sporadic form, you guys, is probably the most common, 85%. And this is where, for some reason, in some of us, as we age, and very important, you guys, this is an older person's disease, our prions spontaneously misfold. Now, there might be environmental triggers. We just haven't figured it out, right? But this represents the majority of CJD cases is the sporadic form. They have done the amino acid sequence of the prions of the human population. And it seems that if you have a specific amino acid, I think it's like a methionine at position 129. I won't ask you that, you guys, but just so you know, it seems like you're at higher risk for spontaneous misfolding of your prion proteins. But you guys, there's so many people that have that amino acid sequence in their prion protein, and they never develop the prion diseases. So it seems like it puts you at a little bit higher risk, but it doesn't determine that you're going to develop a prion disease. Again, folks, in the sporadic form, it's in older folks, over 50 years. And it's thought, you guys, that it was probably a sporadic CJD that first started the Kuru epidemic amongst the 4A people, Papua New Guinea. Yeah. And then, folks, the heartbreak was that in Britain in the, let me see here, this would have been like the 80s, doctors were having young people your age present with what looked like Creutzfeldt-Jacobs disease. But you guys, if you were a healthcare provider and a young person your age presented and you're like, gosh, this looks like CJD, what would bother you? Too young, right? Yeah, you guys are too young for that sporadic form. So the physicians in Britain started trying to figure out what was going on with these young people. And it was determined, folks, that they had become infected with the misfolded prions by eating what? Beef from cows that were infected with the mad cow disease prion or bovine spongiform encephalopathy prion, right? And at the time, it was about a little less than 10 years prior that mad cow disease, BSE, had evolved in Britain, and people were really worried. They're like, well, is it okay? Is it safe to eat British beef, right? And the agricultural minister said, sure, it's safe. And here, watch me eat this big hamburger made from ground British beef. Of course it's safe, right? And it turned out that he was so wrong, right? Yeah, it's just a heartbreak. Let me just keep going, you guys, to see how we're going to do this. This is just more information, you guys. The questions I'm going to ask you on the lecture exam 3 will be pretty superficial, but it's just so many people are fascinated by these prion diseases. So this is just talking about the different types of the CJD, the Creutzfeldt-Jacobs disease, onset of disease, and symptoms. And you guys, the symptoms will depend on which part of the brain is being infected, right? Can definitely develop psychiatric disorders. A lot of times there's balance problems, right? Yeah. Yes. And again, you guys, you're not going to have to memorize this, but to me it's really fascinating that the different types of prion diseases seem to strike different parts of the brain, and again, that will be reflected in different types of clinical signs and symptoms occurring. This, to me, is a little scary because let's see, what is this graph telling us? So here's years, here's time along the x-axis, and y cases is cases of Creutzfeldt-Jacobs disease per here. So this is the number of people, you guys, dying from CJD in Britain per year, and what's the trend? It's increasing, right? And again, folks, the UK was trying to be really thorough. Whenever there was a cow or a steer or a bull or a calf that was diagnosed with mad cow disease, not only would that animal be destroyed, the entire herd would be destroyed, right? So, I mean, it was devastating. And so they're like, well, you know, if they're wiping out the source potentially of, well, and this is sporadic, you guys, but it makes me wonder. This is supposed to be the sporadic form, the form where the prion spontaneously misfold. But I do wonder, you guys, if some of these CJD cases aren't truly the variant form of Creutzfeldt-Jacobs disease, the form that we acquire through ingesting beef that has a mad cow prion in it. And again, you guys, the concern with prion disease is they can have really long incubation periods. So I'm not convinced this is all sporadic. I'm wondering if some of these increasing cases are people that ate the mad cow beef and have been incubating the prion diseases for a long time, and now they're appearing as if they're sporadic forms. And again, folks, just some details on it. Yeah. So again, if in your gene for your normal cellular prion protein, if the DNA sequence encodes information for methionine at amino acid 129, it seems like you're at higher risk for prion disease, but it doesn't mean you're going to develop it. And this is the reason, you guys. So it turns out when they do surveys, 40%, almost half of humans have that particular amino acid sequence in their prion protein, and not half of us are developing prion diseases, right? So there must be some other trigger that's going on. We just don't understand it. We just don't know. So again, folks, just the bad news with prions. Remember, they are one of the most resistant biological structures known, more resistant than endospores. Will normal autoclaving destroy them, folks? No, normal autoclaving will not destroy them. And when we talk about mad cow disease, and if people are beef eaters, they'll say, well, what if we cook the beef really well? And it's like, that's not going to do it. You'd have to incinerate the meat, right? I mean, it's like, that. What's the point, right? So cooking, boiling beef will not destroy the mad cow prions that can be transmitted to people. And again, folks, we know that, if prions are on neurons, and there's motor neurons and sensory neurons that innervate meat, muscle, we know they're going to be there. We also know that the prions can be carried by white blood cells, so they're rich in lymphoid tissues, right? Okay, as we mentioned, you guys, UV won't destroy the prions, or normal autoclaving. And again, folks, this is the nightmare in hospital settings, is that even autoclaved surgical instruments, if they've been contaminated, right, the prions might not be destroyed, and certainly tissue and organ transplants are a source of prions. And it has been discovered, you guys, that blood can carry the prions. And this is why, you guys, if you go to donate blood here in the United States, they'll ask you for kind of like a travel history where you've lived. And if you've lived in Britain, and I don't know the range, you guys, but if you lived in Britain, like during the time of the mad cow outbreak and maybe before then, they won't take your blood because they're worried you could be incubating the variant form of Creutzfeldt-Jacobs disease. They just don't want to risk it, right? And you guys, again, not to make you crazy, but people are like, well, if normal autoclaving won't do it, is there a way to destroy disease-causing prions? And they have developed protocols, you guys, but you need to remember, a lot of medical instruments are so delicate, they wouldn't survive this kind of treatment. But just to give you an idea, folks, so it seems like soaking your instruments in sodium hydroxide or bleach, and then having repeated or extended autoclaving can destroy, denature the disease-causing prions. But again, you guys, the problem is a lot of medical instruments, this would destroy them, right? And also, I was thinking about, what about like plastics? It's like this would destroy the plastics if you're using any kind of plastic or rubber. I was thinking about, what is it, you guys? They call the endoscopes, right? Like endoscopes. It's like, no, that's probably not going to work for those. So what's really needed is a decontamination protocol that can be used on delicate medical instruments, right? Because not everything can withstand this kind of treatment. So we know we could incinerate, yeah. And indeed, you guys in the UK, when they were killing all the cattle that they could identify that had mad cow, traditionally on farms, you dig a big pit and throw your dead animals in the pit, right? But the concern is if the misfolded prions can survive for years, decades, maybe centuries, then that pit where the animals would bury would be a source of disease-causing prions later, right? Or if scavengers came by and dug up the carcasses and dragged the meat around, right? So with the eradication campaign in Britain, they burned the cattle bodies. And it's just, I was like, God, it's like Dante's Inferno, you know? Some of our friends, one of our friends is a veterinarian in northern England, and his farming community got hit. And there were reports that, you know, when you're in farming territory with a lot of cattle, often you can hear the cows mooing, you know, talking like that. And they said it was so sad after these eradication campaigns because you go into the country in these farming communities and it was so quiet, right? And they said the psychological impact on the farmers and the farming family was just horrific, having their cattle destroyed. Oh, I don't want to think about it. All right, so the European Union, we could argue maybe, is a little bit more advanced than us. They have developed enzymes that can help remove the prions from contaminated medical instruments. But as far as I know, you guys, those enzymes are approved in the EU, but they haven't been approved in the United States. With regard, you guys, to prion diagnostics, remember the problem is there's no immune response. So there's no antibodies we can detect to tell us if we or some animal's been infected. So a lot of times you have to wait for clinical signs and symptoms to develop. In humans and animals, behavior changes, right? So there can be psychiatric abnormalities. There can be troubles with balance. In the Kuru documentary, guys, you can see the poor folks. They're just like, you know, can't stay upright. People say sometimes it seems like people that are being affected are kind of laughing hysterically, but I have a feeling it's not out of joy. This uncontrolled laughter, I have a feeling it's just the opposite. They're going through really horrible experiences. Historically, you guys, it would be after the person or the animals died, you would take sections from the brain, right? You would cut them in really thin sections, stain them, and look at them under the microscope to look for the holes in the brain, so histopathology. But that doesn't help us, right? But, you know, when the person's still alive, the animal is still alive. For the ingestive form of prion diseases, you guys, they'll take tonsil biopsies because the tonsils represent like lymphoid tissue, and sometimes they can detect the disease-causing prions in tonsil biopsies. And they'll do that with cattle. They'll also take lymphoid tissue, guys, and this was like for scrapie here in the United States. They'll take a rectal biopsy, a third eyelid biopsies in goats and sheep while they're still alive to screen the flock for the presence of the scrapie prion, right? And if, again, they identify an animal that's infected, they'll wipe out the whole flock. And they'll try to track back where did that animal come from, and they might track back to the herd of origin, and they might screen them and eradicate them. So this is grim stuff, you guys, really grim. Again, folks, I was trying to update this last semester on do we have any better, what they call ante-mortem diagnostic tests before death, right? And so this was one paper I found that they have a skin test that they show that you could, and these are in lab rats, right, rodents, but they were able to detect the misfolded prions in rats they had inoculated. They could detect the misfolded prions in skin samples before clinical signs developed. So again, the hope would be that we could use this for humans and also for meat animals like cattle, sheep, goats. And this is there's one heartbreak after the other. So far, there's been no successful treatment developed, right? So they were experimenting with all kinds of drugs. They were using quinacrine, an anti-malarial drug. Chlorpromazine is an antipsychotic drug. And in lab animals, it appeared like it maybe slowed down the progression, but it did not cure. It did not prevent the fatal disease from developing. So it's like, gosh, you guys, we just need like one of those. We need somebody that breaks that envelope of our understanding, right? To try to get better therapy to treat people that are infected. And man, it would be so good if we had some kind of vaccine. Right. All right. Okay, and you guys, again, I was getting kind of nerdy here, but again, I was just trying to update my own understanding of what was being done to try to combat the prion diseases. So if you're interested, this was an attempt to use antibodies against the prions. But again, you guys, kind of nerdy. I probably shouldn't even have put it in there, or I should have moved it to the end of the presentation here. We do want to remember, folks, that prion diseases are examples of proteopathies, protein misfolding diseases, and other proteopathies includes Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, Lou Gehrig's disease. So really fascinating area of research, you guys, use protein misfolding diseases. With the scrape, you guys, I'll try to go through this quickly. We can see, folks, that in the scrape-infected sheep or goats, the farmers would observe them. It seemed like they were scraping themselves as if they had an incurable itch, right? And maybe that is true, you guys. Maybe the animals do feel like they have this incurable itch, so they keep scraping themselves against a rock, a tree, a fence, a building. But if you look at the Kuru documentary, you can see, folks, the humans are totally off balance, you know? They're just going like this. And so another thought is maybe another reason that goats and sheep are scraping themselves along a fence or a barn is they're just trying to keep upright, yeah? But you can see here, folks, that they scrape themselves, they damage the skin, and then they bleed on those surfaces. And again, the prions are in the white blood cells, so then those surfaces become contaminated with the misfolded prion. So if another healthy animal comes along and does have just a good old itch, right, and maybe damages their skin, those prions could gain entry. This is really bad news, you guys, because they determined that if a female sheep, a ewe, gives birth, or a female goat, a doe, gives birth, the misfolded prions are in high concentration in the placenta and placental fluids. And you guys, sheep and goats are really curious, so if somebody gives birth to a baby, you know, the flock mates will come around and lick the baby, maybe chew on the placenta, you know, nom, 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 yum. And of course, that's a way for them to get infected. And furthermore, you guys, what about the pastures or the grasslands upon which those animals are living? Like, would these grasslands and the soil become, I would say, almost like permanently contaminated with disease-causing prions, right? So in ingesting grass that's been contaminated with the misfolded prions, it seems like, gosh, you know, that's another way that they can be passed on. So what a just really troubling disease. They found prions in milk and colostrum. Colostrum is the first milk that mammals make. It's chock full of antibodies. And this does make sense, you guys, because we don't think about it, but milk has white blood cells in it, right? Okay, and then the heartbreak, you guys, we probably mentioned this already. So historically, if you were a farmer and you had a sheep or a goat that was sick, you can't slaughter sick animals and introduce that meat into the human food chain, right? Or if you had a sheep or a goat that died on the farm, that meat can't go into the human food chain. But what you can do is sell their bodies for salvage. And salvage involves boiling, cooking the bodies, and producing a really high protein feed additive called blood and bone meal. And so the heartbreak was, you guys, that the sheep and goats that had the scrapie prion, their bodies were made into this high protein blood and bone meal and added to cattle feed. And we think that's how the scrapie prion jumped into cattle. And then we had a brand new prion disease, mad cow disease, or bovine spongiform encephalopathy. So the prion jumped from sheep and goats to cattle, and then from cattle, the prion jumped where, you guys? Into humans, right? So you guys do not trust prions. Do not turn your back on a prion, right? And the reason I say this is we're going to see another prion disease we have here in the United States. And people say, oh, it's not zoonotic. What does zoonotic mean? Transmissible from non-human animals to humans. And I want to tell you guys, don't trust prions, okay? So we'll keep going here. And so, folks, this was just to give you an idea of The scrapie, these are flocks of scrapie-infected goats and sheep. This was like 16 years ago. And in the United States, we have a scrapie eradication campaign, just like UK had a mad cow disease eradication campaign. And again, you guys, what it means is you find one animal in a flock, you kill the entire flock, and you even track back to see where that animal's born. If it was born on another farm or another flock, and you might wipe out that flock. So you guys, and again, this is horrific work, but they've reduced the flocks. Let's see, in 2005, there was over 100 flocks here positive in the United States. And I just checked it last week. And I think we're down to like 3 or 4 flocks that are still positive, right? So, I mean, they're making progress. But man, you guys, that eradication work is just horrific work. Yeah. So again, you guys, just showing that here in the U.S. through the eradication campaign, we are really reducing the sheep and goat populations that are positive for scrapie. Okay, so now you guys, we're going to move to cows or cattle and BSE, which first appeared in Britain in 1987. And you guys, you know I have a very weird sense of humor. So here are 2 misfolded prions, and they're talking to one another, and they said, we could never have done this without the humans, and what the prions are talking about is the new disease, BSE, considered an emerging infectious disease. So we want to see what part we humans played in the emergence of bovine spongiform encephalopathy. And folks, I know you can read, but this cartoon still blows me away. So I hope you'll bear with me while I read to you from it, because I had no idea where these salvaged animal products end up, right? And remember, you guys, the concern is that if any animal goes through this salvage process, if they are infected with a disease-causing prion, the cooking process isn't going to destroy them. And now we want to find out where those products end up. Okay, so folks, it goes, this is from the World Health Organization, bovine spongiform encephalopathy in the United Kingdom. So you guys, so here's our farms, right? Here's our farms. Here's our ruminants, you know, cattle, sheep, goats are ruminants. Here's our non-ruminants, like pigs and poultry and fish. So you guys, so far there's been no prion diseases identified in non-ruminants. So as far as we know, pigs don't get it, poultry don't get it, fish don't get it. But the big one in ruminants. So the great recycling. From horns to tails, industries recuperate everything. Many products find their way back into the food chain, human and animal. Safe? Not so sure. I'm like, not so sure. No, it's not safe, okay? So even for cosmetics or medical products, the threat from prions cannot be disregarded. So you guys, so here's the slaughterhouse, okay? So if an animal is healthy or if an animal is incubating prion disease, their tissue then can be sold for human consumption. Animals that are sick, animals that have died, or parts of the animal that humans won't eat, goes into this big salvage process, right, where we take all those animal products, we cook them up to make these animal extracts. Now, where do those animal extracts go, you guys? Let's check it out. So salvage, knackers is the British term for the people that do the salvaging. Knackers process non-animal remains, bones, skin, blood, feathers, rind, offal, as well as bodies of animals collected from farms. The waste is mixed and cooked using methods which are not always guaranteed to be effective against prions. You guys, they are not effective. I mean, they're kind of wibble-wobbling here. No, cooking will not destroy the prions, period. Where do they go? Millions of tons of animal waste recycled each year. Various extracts, elastin, collagen beauty creams, glycerin in toothpaste, lipstick, some soft drinks, keratins in shampoo. Did you have a clue? I didn't. Tallow is fat, animal fats, omnipresent, means it's everywhere in foodstuffs. Biscuits, and I think biscuits, you guys, is the British term for what we call cookies, my favorite food group. Ice creams, stock, broths like soups, cooking oils, also used for making soap. Gelatin, from sweets to pâtés, desserts to medicines, even for the preparation of table wines. Jello gelatin, you guys, when I was growing up, Jello gelatin was like the biggest treat. And what is it? Yeah, okay. Animal feed. Millions of tons are produced each year. So they add these protein supplements, the blood and bone meal, you guys, to animal feeds, right, is a cheap source of protein. Millions of tons are produced each year, prohibited for cattle and sheep, reserved for pigs and poultry, and also fish, for which it is presumed there is no risk. Would you trust a prion? Never, right? To me, this is just really kind of disturbing. And furthermore, folks, the problem is, let's say you've got a feed mill that is making ruminant feed and is also making cattle and poultry feed, right? It's like, how can you run, say, a batch of pig or poultry feed to which you can add ruminant protein, right? And then make sure it's clean enough so that when you run a batch of cattle feed, you know that the cattle feed's not going to get contaminated. It's just like, gosh, boy, it's just, right? Anyway, that diagram to me, you guys, just blew me away. I was just like, oh, amazed.
So you were saying earlier, though, that the chicken and pigs still have not had...
Yeah, there haven't been any prion diseases identified in pigs, in birds, or in fish. Yeah, and so ruminant protein supplements made from ruminant bodies can be added to pig and fish and poultry feed, right? But to me, it's like, that's probably not even a good idea. Yeah. So you guys, and I know you know this story, and you're not going crazy. I couldn't find a nice cartoon showing how the prion jumped from sheep into cattle and then to humans. This, I think you guys, I'm not sure, this looks German to me. Okay, but anyway, so you're not going crazy, right? So we have the scrapie prion, the scrapie prion found in the blood and protein added to cattle feed. The scrapie prion jumps into cattle, binds the cattle prions, causes them to misfold. Now we have a BSE, a mad cow prion, right? And then the big heartbreak was that the cattle tissues that were infected with the disease-causing prions, when they were consumed by young people, right, like you, the BSE prion bound to the human good prion and triggered them to misfold, right? So now we've got this brand new, what they call variant Creutzfeldt-Jacobs disease. So you guys, if we were to watch the prions jump, so the scrapie prion jumps into the cattle, causes the cattle prions to misfold. The cattle tissues are eaten by young people. And we end up with what's called variant CJD. Because remember, you guys, the sporadic form, it happens when we're older. The variant form comes when you eat beef with a BSE prion. So truly an infectious form. And folks, the reason we think young people got hit so hard with it is if you eat beef, folks, and you don't have much money, are you buying beautiful, expensive steaks? No. What are you buying a lot of times? The ground beef, right? And the ground beef, guys, one of your colleagues was a butcher, and he goes, oh yeah. So what we do if we had like a beef carcass is we work on it all day. We'd be trimming things and parts that maybe people didn't want to eat or didn't like to look at. And he goes, at the end of the day, we took all those parts, those trimmings, and we ground them up and made our ground beef. And so a lot of the parts, you guys, they could contain, for example, this is horrible. I'm sorry, you guys. I'm going to make you really upset. But when cattle are killed commercially, they use bolt guns. They hold the bolt gun up to their head, and under pressure, a bolt is driven into their brains. And they've done tests. Brain tissue, guys, is found throughout the neck down into here. There's so much pressure when that bolt is charged. So you have brain tissue blown into surrounding tissues. And you know, this is going to be, these cuts are going to be used for beef. So young people are at higher risk because they're eating the ground beef that had a lot of tissues that potentially we're going to have high concentrations of prions. So you guys, don't eat spinal cord, don't eat tonsil. And you're like, why would we eat tonsils? Well, what about beef tongue? Isn't beef tongue kind of a classic delicatessen, right? And when you cut out the tongue, there's a chance you can catch tonsil, right? And folks, intestines. Now you might say, well, we don't eat intestines. But folks, what do you think is the casing for hot dogs and sausages? It's intestine, right? And intestine is chock full of lymphoid tissue, right? So you know, it's how should I say this, guys? I don't want you to be afraid. However, if we were to have an outbreak of mad cow disease, say in California, I mean, me personally, I would stay away from beef, or if you're gonna eat beef, stay away from brain, spinal cord, tongue, and any kind of sausage or hot dog, right? So, I know, isn't this horrible? Following the mad cow outbreak, like when it was going on kind of hot and heavy, I did stop eating beef, and then slowly I started eating a little bit more, but I have definitely cut way back on how much beef I consume. And I'll tell you why here in just a little bit. Okay. So again, folks, we just see the jump of the prions from sheep to cattle to humans, right? And so folks, fortunately, finally, laws were enacted to prevent feeding of that ruminant blooded bone meal to other ruminants, right? So the ban was not to feed cattle, sheep, and goats mammalian tissues, right? And then 2008 prohibits inclusion of brains and spinal cords of animals older than 30 months in any animal feed. Yeah, this troubles me, guys, and sorry to go off on this, because they're like, okay, well, we'll prohibit inclusion of animals older than 30 months, like cattle older than 30 months. We won't let their bodies be used to make feed supplements. But the problem is, you guys, a cow could get infected when she's a calf and have this really long incubation period. She might not have clinical signs or symptoms of BSE, but still be incubating the prion disease. And so this troubles me that it's like, to me, misleading the public that, oh, we'll only use young cattle, for human beef or to include in these supplements. And it's like, but the prion could still be there. That's just my little nerdy thing. Okay. And again, folks, the concern is there's a long incubation period for cattle prions that might be almost 3 years before they present with clinical signs and symptoms. And so the government is saying, well, if we kill them before then, we should be fine. And it's like, okay. Now, this one, folks, I think is really important because we have our very own North American prion disease of deer, elk, and now they've discovered moose can get infected. And this disease is called chronic wasting disease. It originated in Colorado. So it originated in Colorado and is spread north up into Canada, south. It's spread west, but it hasn't gotten to California yet, folks. And you might say, how did it jump, from these infected states to West Virginia and New York? Well, this is because there is, and you guys might not be familiar with this, but there are game ranches where, like, deer, elk might be raised, and then hunters will come and hunt them and shoot them, right? And you guys, I want you to know, my mom was raised in Colorado, and elk and deer meat was what her family survived on, so I come from a hunting family. But anyway, I better shut up. Okay, anyway, right. Okay, so the reason we see the chronic wasting disease jumping is that these animals are transported. They'll be transported from one game farm to another. So again, kind of the humans involved in helping them to spread. So the reason it's called chronic wasting disease, folks, as you can see, this animal appear emaciated, right? They stop eating, they waste away. Again, it's elk, deer, and now moose. And my concern, you guys, is that not only do humans eat, deer, venison, elk, and moose, and so could it be zoonotic? They keep telling us it's not, but I like I said, I never trusted prion. And furthermore, you guys, a lot of the deer and elk and moose, they graze the same areas that our beef cattle would graze. And so I'm like, well, couldn't we get, the same kind of jumping going on? Could we get a jump of chronic wasting disease going on into our beef cattle, and then we'll have our own unique form of mad cow disease? Or, you guys, just directly, could there be a jump of the chronic wasting disease prion into humans, just as we saw with the mad cow disease? So they do screen, like, during hunting season in these areas. Like, if you're a hunter and you're coming back with a carcass, maybe tied to your roof, something like that, they have inspection stations, right? So they'll inspect to see if the animal looks like this, they're probably going to confiscate the carcass, right, and run tests on it. As a hunter, you can submit lymph nodes from your carcass and have it screened for chronic wasting disease. But a lot of hunters are like, well, you know, a deer carcass is like 200 pounds of good venison, and it's like, they tell me it's not zoonotic, so I'm still gonna eat it. And I don't know, you guys, it just really troubles me. It seems, I don't know, I think if my family was still hunting, and they were still hunting elk and deer, I think I would say, I would still eat it, you guys, as long as we send in a lymph node for them to analyze. I would not eat it if it came back chronic wasting disease prion positive. I just don't think it's worth the risk. But this is becoming a big dilemma in many of these states where hunting is really important. Okay, and this was a chronic wasting disease, you guys, in 2018. It is definitely spreading, and there's an article, I think I got it posted, you guys, on Canvas, that they think some deer herds are going to be totally wiped out from chronic wasting disease. And again, you guys, my concern is contamination of the grasslands where these animals are sharing space, you know, with one another and with domesticated cattle. Other TSCs, you guys, cats can develop it, mink can develop it, squirrels, and you're like, squirrels? Well, and I think, folks, in some parts of the United States, squirrel brains is kind of a delicacy. You guys don't eat brains. No more brains. No, seriously. And I know that's really hard because if you visit somebody and they offer you food, right? It's so rude to not eat the food, but if you guys try not to eat the brains. I don't know, hide it under the, I don't know what. Don't eat brains. Not good for you. Okay. And folks, just with that kind of wild and crazy thing, I think that's going to be it. Because this, oh my God, it could keep going and going and going. I just put, again, folks, just these last slides, I was trying to find some updates on diagnostics, also on normal prion function, and most of the papers were seriously over my head. So I provided some links there if you're interested. But I think we're going to leave just with the caution of you guys, don't ever ingest a misfolded prion readily or voluntarily, right? Yeah, okay. So folks, oh, one thing, I mean, another concern that people have had is with dental procedures, or like, what if a dental procedure is being done on a person that's incubating a prion disease, right? And so then it's the same concern over dental instruments, right? Of course they're going to be cleaned and sterilized, autoclaved, right? But do those dental instruments then become a source of disease-causing prions? And you know, sometimes, folks, we just can't think about it. Sometimes you just have to say, okay, that's not going to think about that, all right? Because we need to go have our teeth cleaned and have those dental protocols done.
Those on the chronic waste.
Pardon, say again?
On the chronic waste.
Yes.
So I actually just moved back to Colorado. I'm an archery hunter out there with elk and deer, and it's crazy to hear about that because I mean, they Department of Fitness, not fishing, it's Colorado Wildlife thing out there.
Yeah.
They mention chronic waste disease, but they don't obviously educate you as much on it. But I know hunters, they go out and try to be more, where you do everything yourself, you create your meat, you grind it and everything.
Yes.
And they don't get, I know a guy that does not get his meat tested at all. He goes out and kills his elk and he goes home and he starts grinding it and cutting it himself.
Yeah.
No testing whatsoever. So it's
It is a little bit crazy, yeah. And the article that was written, it was written. Oh gosh, I forgot the name of the magazine, but it was written by a husband and wife team, and they have a daughter, a little girl, and they said, you know, before they were willing to kind of risk it, but now the whole point was, do we risk feeding it to our daughter? And so they were going to have their elk and deer tested now, because now they're feeding it to their little daughter, right? Yeah, boy, it's a tough one, you guys. Well, I know what I wanted to share with you. It's like, well, what about mad cow disease here in the United States? So we have had some cases of mad cow disease here in the United States. The first one, this is just my twisted sense of humor, you guys. It was in Washington State. They identified a cow, and she had mad cow disease. But there was jubilation when it was discovered she was from Canada. And this was about the time that the South Park movie came out, you know, the Blame Canada. You guys are probably too young for that. But I was just laughing, because like, oh, it was all Canada's fault, right? It's all Canada's fault. But since then, you guys, we've had in Texas, we've had in California, We've had the last case of mad cow disease we had here in California. They determined it wasn't that the cow had been fed, contaminated feed or blood and bone meal contaminated with prions. This was scary to me, you guys. They determined it was sporadic BSE. What does that mean? It's the same as sporadic CJD, that in those cattle, some of the cattle, for some reason, their prions are misfolding spontaneously, sporadically. And I'm like, that is such bad news, again, for us. And it makes me, I support ranchers and farmers. I'm a meat eater. But man, I am really going back to that. I think I'm really going to be cutting back on my consumption of beef because we don't have a really good testing. We don't test a lot ahead of cattle that go into the for human consumption. In Japan, you guys, they test every cattle carcass that will go for human consumption. Nothing goes into the Japanese beef market unless it's been tested. Here in the United States, I think we test like 0.1%. Anyway, I know that cattle farmers and dairymen are having a rough go of it, but I don't know. Yeah, I will still eat beef, but I'm sure cutting back on my consumption of it. Oh, and the other thing, you guys, and apologies, I'm rattling on here. What helps if the cattle have not been fed feed supplements, right? If they are raised how they were meant to be raised, grazing grass, that's going to reduce the chance that they can get infected through these protein supplements. So my thought is, well, maybe if I eat beef, I should be willing to pay a little bit more to get grass-fed beef. And I don't know if that's a surefire way to prevent infection. But okay, I'm rattling now. Sorry, guys. It's the end of the semester. I rattle more and more as I get towards the end of the semester. Shall we move on? Now that we're all cheered by the thought of prions everywhere. Okay, so you guys, this is like the second to the last big unit for the course, and it's called medical microbiology. And indeed, folks, there's kind of like two sections to this, and if we were doing it chronologically, I would do what's called epidemiology first. We would talk about the sources of microbial pathogens called reservoirs. We talk about transmission, how the pathogens leave the reservoir and reach us, the new hosts. But you guys, to me, that stuff is a little bit easier to digest. And you guys, next week, we have the lab exam on Tuesday, and then we have one lecture, and then next Thursday is Thanksgiving, so we only have one lecture next week. So what I wanted to do is give you the hard stuff at the front. And the hard stuff, you guys, a lot of it is going to be information like covered in the virulence factor worksheet. Did I pass this around to you guys? Okay. And the toxin table. And my thought is the longer you have to think about it, the more time you've had to process it, and then you'll have a better grip on it for lecture exam 3. So we're going to kind of dive right in and talk about virulence factors. What is it that microbes make that let them invade us, outwit our immune system, and cause harm, right? So this will be the first PowerPoint we look at. And then the second PowerPoint that I'll post is on the epidemiology. What are the sources of the microbes? What are the different ways they can be transmitted to us? Because that stuff isn't as intense as this virulence factor information. Okay, so we're kind of doing it cart before the horse. Okay, so this then is another way of thinking about this medical microbiology is how to be a good pathogen. And the reason is, folks, that if we look at all the microbes in the world, most of them do not cause harm, right? Most of them are either free living, or most of them, maybe they live with us, but they don't cause harm. They're commensals, or many of them help us, right? So this is an unusual behavior to cause harm. So we want to take a look at, if we were a microbe and we wanted to be a virulent pathogen, what is it we would have to do? What is it we would have to make to be able to cause harm to other organisms? So kind of a background, folks, it's a historical background, is even though you guys are probably taught from when you're just little tiny kids about the germ theory of disease, like your parents teach you to wash your hands, And if maybe one of your friends is sick, your parents might say, well, let's not play with your friend until they're better, right? So you guys probably grew up almost instinctively knowing this germ theory of disease. And the germ theory of disease, folks, states that a microbe, a germ, can invade a host and cause harm, right? And you're like, well, sure, right? But folks, until the 1800s, the germ theory of disease actually wasn't proven. And the reason was, even though some folks would say, well, okay, we agree that microbes, germs can cause disease, we believe that those microbes, those germs, can spontaneously arise inside a person because they aren't good people, they're bad behavior. You know, they've done something wrong. You know, they've maybe offended God. Who knows, maybe they're a witch or a wizard, right? So even when people were convinced that microbes could cause infectious disease, a lot of people still thought the microbes just arose within you if you were a bad person, right? So to prevent the transmission of disease, it was really important to prove that these disease-causing germs come from outside of us, right? They come from the outside and invade us. And only when we understand that and accept that can we then work on preventing the transmission and spread of diseases. So this was actually a big deal, you guys. The two people that played a crucial role in developing the germ theory of disease and then proving them were good old Louis Pasteur and Robert Koch. And some people say that Louis Pasteur was really important in developing the germ theory of disease. And it was fascinating, you guys. He was working with the French wine industry. The French wine producers couldn't figure out why their grape juice was turning sour, right? Instead of turning into alcohol, wine. And Pasteur discovered, well, the grape juice was contaminated with a bacteria that carries out fermentation of sugar and makes acids. That's why it was going sour, right? So he developed pasteurization. You kill the bacteria by heating the grape juice, and then you add your good Saccharomyces back. So he saved the French wine industry. So he talked about this bacteria contaminating grape juice as diseases of wine, right? It was described as a disease of wine. A pathogen causes disease of wine. And then he next worked on silkworms. So the silkworms were getting sick, and this was destroying the silkworm industry, right? And he discovered the silkworms were dying because they were infected with microbes. So Pasteur was really important in developing this concept that microbes can invade other organisms and cause disease. The problem was proving it, right? The problem was proving it. So Pasteur and Koch, who were contemporaries, they were kind of racing against one another, competing against one another to prove the germ theory of disease. And you guys, Pasteur's lab had a huge disadvantage. They were working with liquid media broths, right? Like soups, right? And it was Koch's lab that developed the use of agar as a solidifying agent, right? So Pasteur's lab, working with broths, they could never guarantee they had pure cultures. In contrast, Robert Koch's lab, working with agar and agar plates, right, they were able to isolate individual colonies, which are pure cultures. They were able to guarantee they were working with pure cultures. And ultimately, it was Koch's lab that Koch and his colleagues that developed 4 steps, 4 prerequisites to prove the germ theory of disease, right? But you guys, I think both men deserve credit for helping to prove the germ theory of disease. Now, Koch's postulates, you guys, again, these are 4 steps, 4 prerequisites to prove a specific microbe causes a specific disease, right? And we're going to walk through these steps, you guys, and probably on the lecture exam, I'd like you to know the basic 4 steps of 4 prerequisites, and I want you to see, you guys, how pure culture is essential. You know, we've made such a big deal in lab about pure culture, right? So pure culture was absolutely essential in proving Koch's postulates. So folks, in Koch's postulates, let's say there's some microbe X, and our hypothesis is that microbe X causes infectious disease Y. Okay, we'll just be generic. So in the 4 steps then, your suspect microbe X has to be isolated from all cases of the disease, right? So all of the animals, all the humans, they get sick with that particular disease. You have to isolate the suspect microbe in pure culture, right? So here we have some sick rodents, We're going to take tissue samples on auger plates. We're going to streak them. We're going to get all the microbes in pure culture. Okay? So we have to grow the suspect microbe in pure culture. That would be step 2. Okay? And then, folks, and this is the ethical part with Koch's postulates, then you have to take healthy animals and inoculate them with the pure cultures of your suspect pathogen. And then your test animals that get injected, they have to develop the same infectious disease, right? So ethically, you guys, this gets really tricky. And then from the sick or dead test animals, what must you do as a final step? You have to re-isolate the suspect pathogen in pure culture to prove that it was that pathogen that caused the infectious disease. So again, folks, Robert Koch he and his colleagues were the ones that developed these 4 postulates, and they're still used today, folks. And again, very, really ethically kind of challenging to purposely infect animals. And certainly, you guys, with some pathogens, you can't do this. What if you have a pathogen that only infects people, like HIV, right? You can't carry this out. And furthermore, you guys, with HIV, since it's an obligate intracellular parasite, you can never truly grow in pure culture, right? You always have to have host cells present, right? So they modify Koch's postulates for pathogens that only infect humans, or say for viruses where you truly can't grow them in pure culture, right? Because there's always going to be that host cell present. So they modify them kind of to make them more flexible, right? But the basics of Koch's postulates are still referred to in modern medical microbiology. And then, folks, another background concept is you guys intuitively know that some microbes can cause more damage than others. Like, for example, you guys, I know this is silly, but if I told you, okay, you have to be infected with a virus, but I'm going to give you a choice. You can choose to be infected with a cold virus, or you can choose to be infected with the Ebola virus. Now, instinctively, you know which one are you going to choose? The cold virus, right? Why? Because the cold virus isn't going to kill you, right? And the Ebola virus, there's a good chance you could die or at least have really serious harm done, right? So you guys, we, as microbiologists then, we want to measure the amount of damage a microbe can do. And the amount of damage a microbe can do, we're going to call that virulence. Okay, so there's some terms that we've been using, guys. So if we use the term pathogen, okay, patho comes from pathos for suffering. So a pathogen is something that can cause suffering. Or we would say causes disease. So if I mention a microbe that's a pathogen, we know, oh, that microbe can cause disease, right? But remember, you guys, a cold virus is a pathogen, and Ebola virus is a pathogen. So again, the measure of the damage, the measure or the extent of damage, we could describe this as virulence. So you guys, if a pathogen has low virulence, what does that mean? Little damage, right? Like a cold virus, right? Little damage, little harm. Maybe makes you uncomfortable. Little damage, little harm. So the perfect example would be like a cold virus, right? You're not going to die from it. But folks, what about high virulence? Lots of damage. And what's the ultimate type of damage? Death. And in our example, you guys, Ebola virus would be an example of a highly virulent pathogen, right? So then, as scientists, you always want to work with numbers. So they were, the medical microbiologists were trying to come up with some system where you could compare, using numbers, compare the virulence of a microbe. And so 2 ways we can compare virulence of microbes, you guys, is by using 2 values. And the 2 values are infectious dose 50 and lethal dose 50. So measuring virulence, the degree of damage, we can use infectious dose 50 and it's usually subscript, so it's ID 50. And then another one is called lethal dose 50, or LD50. So folks, infectious dose 50 is the number of microbes required to infect half, 50% of a test population of organisms. And I know that sounds kind of awkward, but you guys, let's say, this is horrible. Let's say we're running this experiment, and pretend, a mind experiment, you guys. Let's say we have a pathogen, and we have multiple sets of 100 people. And the first set of 100 people, we're going to inoculate every person with 10 microbes. And let's say, I'll make this up, guys. Let's say 1% of the people get infected. And then in our second experimental group, we're going to inoculate 1,000 microbes into every person, and half the people get infected, right? So the ID50 for that pathogen would be 1,000 microbes, 1,000 pathogens, right? It takes 1,000 pathogens to infect half the test population. So you guys, this is something that's determined in a lab, and again, a lot of times you can't determine it in a lab because it's unethical, right? But you guys, if infectious dose 50 is the number of microbes it requires to overwhelm the host defenses to infect half the test population, what do you think LD50 is? The number of microbes required, right, to kill half the test population, right? Now, folks, this is where it gets a little bit weird, kind of like those inverse relationships that I always have to kind of stop and think about. So, folks, what we want to remember is there's an inverse relationship between ID50 and LD50 and virulence. So, you guys, as the ID50 or LD50 decreases, what's happening to virulence? It's increasing, right? And folks, if we think about this, like for a microbe to invade us and cause disease, it has to outwit our natural defenses, right? And so if you have one microbe, and you guys, I'm going to make this up. Say microbe A. We have microbe A, and let's say the ID50. We'll put it here. And then we have a microbe B. And let's say the ID50 for microbe A is 10,000 microbes. Okay, it takes 10,000 microbes when you infect your test population of animals to cause half of them to get infected, right? It takes 10,000 microbes to overwhelm the host defenses. And in contrast, you guys, microbe B, let's give it a really low ID50. Let's say it's 10 microbes. It takes 10 microbes to overwhelm the host defenses. So which microbe, you guys, is more virulent, A or B?
B.
B. My gosh, you guys, this means a single microbe has the ability to overwhelm our natural defenses, which we're going to see. We have so many natural defenses, right? So with a low with a low ID50 or LD50, right, that means that's a highly virulent pathogen, right? The higher the ID50, the higher the LD50, the lower is the virulence. That means it's going to take billions and billions to overwhelm the host defenses and cause serious harm. Does this sort of make sense, you guys? Okay. So, this was from another textbook, you guys, and what they did is they ranked. Let's see here. Most of these are bacteria. There's one fungus in here, but they ranked some pathogens. And the reason I wanted you to know this, we have all of them here in California, from most virulent to least virulent. And folks, again, we have some of these highly virulent pathogens here in California. Francisella tularensis is a bacterial pathogen, and it causes what's called rabbit fever. It's named tularensis because it was discovered in Tulare County here in California, right? Highly virulent. Yersinia pestis causes bubonic plague, right? Here we have necrosis from bubonic plague. Bordetella pertussis causes whooping cough. Pseudomonas aeruginosa, truly an opportunistic pathogen, causes infections in burn patients, cystic fibrosis patients. Clostridium difficile, this is usually in folks that have been on broad-spectrum antibiotics. And then we get down, you guys, to Candida albicans, another opportunistic pathogen. And then we get down to our lactobacilli diphtheroid. These guys I would consider like commensals, if not outright mutualists. So it's going to be unusual situations where they're going to cause harm. But folks, if I asked you to predict on which end of the scale would we have microbes with the lowest ID50 and LD50, would it be down here or up there? Yeah, up here. The most virulent ones, you guys, are going to have the lowest ID50 and LD50. So be prepared, you guys. I will ask that on lecture exam 3. And again, it takes a little bit of thought. Kind of that inverse relationship is always a little bit challenging. This, you guys, because we were the site where tularemia was discovered. I just stole this off of a internet PowerPoint. Flu-like symptoms. One thing, you guys, with the ID50 for Francisella tularensis, it's so low. It's been one of the a great candidate for a biological warfare agent, right? So this is on kind of the biological warfare list because it takes so few pathogens to infect us, and it can be airborne. It can be transmission through, butchering an infected animal. It's vector-borne. There's all these different ways it can be transmitted. All right, and then, folks, we just have one minute. So, folks, what we'll do is just get started. So now we've kind of talked about principles of the germ theory, how to prove a specific pathogen causes a specific disease. We've talked about how to compare virulence. And then what we'll do, folks, is this next part will be to see how the pathogen, once it reaches us, how does it colonize us? How does it evade or avoid our immune responses? And then how does it cause harm, right? And again, folks, if maybe, we've got a lecture Thursday, good. So we'll get through most of this, you guys, on Thursday, and that will put us in a good setting. Yeah, okay, very good. So you guys, we'll keep going then on Thursday on this first PowerPoint. Okay, so let's see here, half of you I won't see, the other half I'll see you at 1 o'clock, okay?